Hi, everybody. I'm glad you're here tonight because it's such an exciting speaker. This is Julia Douglas, and it's her second time speaking to us. She was so interesting the first time, so we asked her again. And she grew up in Honolulu and is currently now working on her PhD in the botany department at the University of Hawaii. She studies the ecology of epiphytic orchids. I hope I pronounced that right.
Yeah.
And she's currently living in Mexico for a year on a Fulbright scholarship. And she is working with the community in Oaxaca. It's spelled Oaxaca. She's on an orchid restoration project. She loves interesting vegetation, cloud forests, surfing, and ceramics. Mm-hmm. And tonight we have from Mexico, the Canopy Ecology of Epiphytic Orchids in the Osa Peninsula in Costa Rica. So Julia, we're really excited to hear what you have to say. Thank you.
Yeah, thank you. I'll go ahead and share my screen. Okay, can you see it?
Yes.
Okay, yeah, stop me at any point if you can't hear me or it's not working anymore. Okay, well, thank you to the Honolulu Orchid Society for having me again. I'm really excited to speak to you folks and excited to participate in person, hopefully when I come back home in a year. And so I'm going to split the presentation today up into two sections. The first, I'm going to give kind of an update on my project in Oaxaca here in Mexico. And then the second half, I'll talk about my work in Costa Rica. And so both of these elements are chapters of my PhD. And so originally I started with working in Mexico, but I wasn't able to go for a few years during the pandemic. And so while I wasn't able to go, I kind of developed this other project in Costa Rica and worked on that in 2021. And so that the field work for Costa Rica is entirely done. That was all last year. But now I'm back in Mexico right now working to finish the restoration project. And so, yeah, I'll start first talking about the biocultural restoration of Prosechia carwinskii in Oaxaca. And so this is the species that I'm working with in Oaxaca. Some of you might remember from when I talked a year or two ago. It's called Prosechia carwinskii in the Orchidaceae, and it is an epiphytic orchid. And so epiphytes are plants that live on the substrate of trees in the canopy, but they're not parasitic. And so they don't use any of the carbohydrates or any of the nutrients or take anything from the tree. They're just using it as structure for them to grow on. And so many of the orchids that you folks grow are epiphytes, even if they're rooted and you have them in pots or hanging or in kind of different types of medium. In the wild, they would be growing epiphytically on a tree. And so this is the orchid that I'm working with. It's called La Monjita is the name that people call it there. And it grows in the pine oak forests of Oaxaca. And Oaxaca is a state on the southwest coast, kind of southwest area of Mexico. It's mountainous with mountains up to 10,000 feet tall and has lots of pine oak forest where this orchid lives. And so the specific town that I'm working in is Villa de Zachila, which is in kind of the central valleys of Oaxaca. And I'm working in this town because it has a long tradition of using this orchid traditionally to adorn and use the flower in celebrations. And so because of that, I'm focusing on this specific species orchid or orchid species. I know you folks call it a species orchid, but I might call it an orchid species in this town. And so I started this project 4 years ago and have since been 3 times to kind of work on it through the different years. And so it's used traditionally, so people harvest the pseudobulbs, so those storage bulb structures and the flowers during the spring, so that it only flowers in the spring. And so local people go out into the forest and collect thousands of these orchids to decorate their town. And so up to 10,000 have been reported to be taken from the surrounding forest. But it's because there's been such a heavy harvest combined with climate change and other land use changes, the orchid is in decline. And so we really need to think of conservation strategies if we want to prevent the extinction of the orchid. But it's important to also remember that this cultural tradition of orchid harvest is very important to the people of Xochitl and Oaxaca. And so any kind of strategy in which you just prohibit the harvest of orchids would be unethical and disrespectful of this tradition. And so an effective strategy has to kind of work with the people of Xochitl and allow for the continuation of some amount of sustainable harvest, hopefully. And so I went back in 2022, earlier this year in the spring, and documented and participated in a lot of these orchid harvesting trips. And so I went with orchid harvesters and then kind of took pictures of how they were using them. And so then in these Semana Santa or Easter parades, thousands of people will have the orchids in their sombreros with they call it pastle here, or Tillandsia isnoides, or Pele's hair, we call it back home. And so they make these adornments of the Pele's hair and the native orchids. They also then use it to make these offerings and elaborate decorations on the altars of their neighborhood churches. And so thousands of the orchids, you can see them with the pseudobulbs, are used for these adornments. So this last year, 4,000 about I counted in the different churches of this town in Oaxaca. And they're really beautiful adornments. You can see them hanging from the outside of the churches, and people go around during Semana Santa and pay their respects to each of the churches. And so it's at this point a Catholic tradition, but it was the use of this orchid for ornamentation is pre-conquest and pre-Columbian. And so they have them hanging from the ceiling on these different types of other native plants laid out on the ground. And they also have a very strong fragrance, this kind of citrusy fragrance. They're in the Cattleya Alliance. And so it's a really amazing, strong fragrance that they have, and that churches are filled with them during the celebrations. And so to work on conservation strategies, earlier this spring, we held a series of meetings with the traditional orchid harvesters of the town. And so I work with the municipio, or the small town government, and we kind of called a meeting. And so this is the group of concheros, or orchid harvester representatives, that showed up to talk about conservation of this plant. And they're all really enthusiastic about participating, because they have noticed throughout their lifetimes and their grandfathers' lifetimes the slow decline of the number of plants that they can find when they go out into the forests and try to harvest them. And so people are interested in conserving the plant as long as the conservation does allow them to continue using it for their religious and ornamental purposes. And so it was really exciting that this many people showed up to talk about it back in March. And so the conservation strategy we've identified is reintroduction, because the way that the people harvest the plant is that they'll twist one of the pseudobulbs off with the flower, with the inflorescence. So they take the entire pseudobulb because they want the flower to last longer in the parades and in the adornments. And so because this is the way that they harvest them, there is a shoot meristem, or a bud at the base of the pseudobulb. So this allows us to reintroduce it, or to transplant it after it's used in the Semana Santa celebrations. So instead of just discouraging people from harvesting, we could reuse material that is otherwise discarded, because usually they just either compost or let the pseudobulbs dry out after the parties. Usually they don't reintroduce them. And so yeah, here's some just results from a preliminary study where we showed that 65% of single pseudobulbs were capable of growing. And you folks probably know this from various orchids that you grow, that if you cut off certain sections of pseudobulbs or of the rhizome, that you can create an entirely new individual. And so the different orchid harvesters from the different neighborhoods agreed to donate a lot of their orchids after Semana Santa. So after all those adornments were made and the parades happened and all the thousands of orchids were used, these men from each of the neighborhoods collected their orchids and then brought them to the municipio to donate to the project that I'm working on to restore some of these plants. And traditionally, it's only men who go out and harvest orchids. That's why it's mostly men here, or all men. Some of the neighborhoods are starting to have a few young women join them on their harvesting trip. So maybe it's changing a little bit, but it's a very traditional place. And so 800 pseudobulbs of Persechia karwinskii were donated this April, which is really exciting here. They all are laid out. Oh, so we measured them all and gave them all individual tags and brought them back to the pine oak forest. And so this is the site that I started working in. So it's in Santa Ines, which, as you can see on the map, is just to the west up into the mountains from Zachila. And so these mountains have a pine oak forest that is suitable for this species of orchid to grow. And so we selected for this just first year 15 trees, or it's called audibles in this map. And those were the 15 trees that we attached the 800 orchids onto. And so the way that we transplanted or reintroduced them was to use a wire to strap them really tightly to the trunk of the tree, and then also use a small amount of silicone to really attach them onto the trunk. And so this was recommended to me by one of the orchid researchers here at the university, that you really don't want them to move at all, or else they won't be able to establish their roots. And so using a little bit of silicone might help. But this is still experimental, and so we're still trying to figure out the best way to attach them to the trees without injuring them, and hoping for the highest survivorship. So even at the end of this talk, if some of you folks, because you're experienced in growing orchids, have ideas about how to best attach them, we're open to ideas. And so we would attach 10 pseudobulbs to the lower part of the trunk, and I would climb the tree with my ropes and attach 20 to the higher part of the canopy. And so of the 800 donated, only about 400 were actually in suitable enough kind of condition to reintroduce and transplant. And so this first year of transplanting, we transplanted 450, and that was in April of this year. And so this was the first attempt to see if we can establish a new population and save some of the orchid material after they're used for the ornamentations, and contribute, not solve the problem of a species at risk of extinction, but contribute to the conservation of this species. And so these are some pictures that are very exciting that were taken in a few weeks after post-planting in May, after the first rain had fallen on these orchids. And you can see that they're already starting to grow their roots at the base of the pseudobulb, which is very exciting. And so I haven't been back since May. Then so there's a family of people who are taking care or kind of watching over the site, and they send me pictures once in a while. Sometimes they're kind of blurry like the one on the right, but you get the idea that it's pretty exciting that they are growing roots. And so hopefully they'll also grow shoots and establish full-size plants from these single bulbs. I would be happy with even a 30% survivorship rate, even 10%, like anything more than them just all being discarded and dying would be a success, I think. And then it's also kind of hopefully a symbolic project too, to work with the orchid harvesters and the town. We're all collaborating on this. And even if we don't actually establish a new population of plants, one of the results could be kind of a slightly shifted mindset of the traditional harvesters there to work on conservation strategies and to think about how they can contribute to this. In the past, there was such an abundance of this species that people could really take as many as they wanted, and there wouldn't be an impact. But that's kind of culminated in the current situation of there being fewer and fewer every year. And so the perspective needs to shift, and the tradition really needs to adapt to new ecological realities. But that can't come from an external source like myself or any other kind of external land manager. That has to come from the community itself and from the local people there. And so hoping to contribute to that process. So here's some of the people from the municipio who are helping me. They would come out and reintroduce and plant the orchids. You can see us all holding the various ones as we would plant them. And so this is the family of people who are taking care of the orchids in Santa Ines. Their names are Giovanni, Sara, and Kevin Gaspar Reyes. And this is the sign that we made to put on the site. It says, this is a site of restoration and research of monjitas, or yellow orchids. Please do not cut or take away the plants. Leave them for everybody, take care of them. And so hopefully when I'm going to return in late October to measure survivorship, and really hoping to find some of them doing well. Yes, and to wrap up this section of the presentation, I'd definitely like to say thank you to the Concheros of Zachu, the orchid harvesters, for sharing their traditions with me and collaborating with me on the project. It's taken multiple years to kind of get it going, but it's hopefully an exciting way to conserve this species, which is so beautiful and important. And here's the different groups of concheros or orchid harvesters from every neighborhood going out to find plants to decorate the town. Okay, so moving on to the second section. Okay, so the second section that I'll be talking about is about the ecology of the Osa Peninsula's epiphytic orchids, specifically looking at environmental variables that influence abundance and species composition of the Canopy community. And so, as I said before, this is a different chapter of my dissertation. And the field work for this occurred last year in the summer of 2021. I went down or over east to Costa Rica and worked on this. And so just to geographically orient us a little bit, Costa Rica is in Central America between Panama and Nicaragua. And so it's a pretty thin country with the Caribbean on one side, the Pacific on the other. Also quite mountainous and very tropical, and also with an extraordinarily high diversity of orchids, as many of you might have heard, or maybe some of you have gotten the chance to visit Costa Rica. And the place that I worked was on the western coast, and so the Pacific coast in a lowland forest of the Osa Peninsula. And so this peninsula is special because its forest is really well preserved, and it's one of the largest remaining tracts of lowland wet forest in Central America. And so a lot of this lowland forest type is lost to development or to agriculture, but this area is pretty well preserved. So a really high canopy of 100 to 100 foot tall, 150 foot tall, or 200 foot sometimes tall trees, very diverse with lots of animals and orchid diversity and insect diversity. And so there's a biological station called Piro Biological Station that I got a fellowship to work at through an organization called OSA Conservation. And so I was there for 7 months living and working and collecting information on the orchids there. And the Osa Peninsula is pretty not super inhabited. There's a town called Puerto Jimenez that's right at this tip. And I'm not sure the population, but most of it is forest and a few agricultural areas, not very populated. And so the context for this project is that there's approximately 1,800 species of native orchid, so specific species native to Costa Rica, and about 150 of those are found in the Osa Peninsula. Within Piro, so within the station I was working at, we have about 40 common species, and these all have very wide distribution. So this kind of means that the place I was working didn't actually have a huge diversity of orchid species. It was like there's some places in Costa Rica where you might have 100 species, maybe 50 species at least on a single tree. But I would usually find between 5 to 10 species on a tree, but this made it easier to do large scale surveys of habitat preferences. And so I'll talk more about that later. And epiphytic orchids serve as bioclimatic indicators. And so epiphytes, like I said before, plants that live non-parasitically in the canopy of tall or short trees, because they're not connected to a terrestrial water source, they have to be very, they have to be resilient to these long periods of time, sometimes without water. And so any changes in atmospheric precipitation, so rain, fog, water really, it impacts them directly. And so their abundance and distribution is reflected of these climatic variations. And so as we're looking forward into climatic changes that are already happening as well, orchids kind of serve as a way as a lens through which to study the process of plants responding to climatic changes. And then finally, conservation and management of any type of orchid really depends on understanding its habitat preferences and the environmental variables which are driving the distribution of the species. So how can you can't design a conservation plan that's effective if you have no idea where an orchid grows or what kind of place it likes or what conditions it needs to thrive. And so because there's not very much research done on the high canopies of trees, it's important to first learn a little bit about the canopy, the high canopy, before designing a management strategy. And so the intent of this project was to contribute to that process of learning more about the orchids of the high canopy. And so the main research questions here are so we've got 40-ish species in this area. They live in a variety of places. Here are some drawings I did of different ones. And then the main question then is, why do some of them live in certain areas? And so you can see in this drawing, the Caularthron bilamillatum only usually lives on the highest part of the tree in a dry, sunny spot. That's where you'll find this orchid. In contrast, the Dressel-rella hispidia, I only ever found it in these shady areas on trunks. And so you have these different places where orchids are growing within a single tree. And so that observation leads to the question of what is the composition, the species composition of the community of epiphytic orchids in the canopy of Piro. So first of all, where are these species and what are the species? And then second, what are the variables, environmental variables, that are influencing that diversity and abundance? So first it's figuring out where the orchids are and what the orchids are, which species they are. And then the second part is, why are they there and why have they thrived or not thrived in these various places within a single and then tree, and then expanding that to the forest in general. And so the trees that I was working with in those peninsula are very tall. So these were between 100 to 200 feet tall, gigantic, beautiful trees. Here's a few, like a Sloania in the middle. You can see Marvin, who helped me on the project, in front, and then a Caryocar costaricensis, or an ahu tree, on the left. These are massive trees with a diameter at the base of up to 20 feet in some cases. So kind of redwood or sequoia-esque, but tropical evergreen trees, or deciduous in some cases, but yeah. Okay, so the process of climbing these trees was quite logistically challenging. On the left is Fry, who helped me. His name is Fry Rodriguez, and he helped me a lot firing the big shot into the trees. So you can see him there it's a 10-foot long slingshot, and he's pulling down the cord. And there's a weight in that little yellow bag, and that's attached to a small yellow string, which a lot of it is then in that little cube thing. And so as so we would identify a branch in the tree that looked safe and thick enough to climb and enough to put my whole weight on, and looked like it wasn't rotten, and was also high enough in the tree that I could survey all the orchids. And then Fry would help shoot the line into the right spot. And if we got that thin line over the right spot, 100 feet in the air, then we would then string the thicker red line, which was my real climbing line, up in and over the branch. And so sometimes this process took as much as 3 to 4 hours to get the line in the right spot, because you really want to make sure it's in a safe place. And then with all the vegetation in the tropical forest, it can be very hard to see and to find a place to shoot the line from. And so that was sometimes quite a frustrating process. Sometimes it took more time to get the line in a tree than it did to climb the tree and survey the orchids. So once the line was in the tree, we use this series of gree-grees and knots and slings to create an anchor on another tree nearby. So the rule was that the anchor tree needs to be bigger than your leg-ish. So something about this wide, and then you wrap it with the slings and the carabiners, and then have this system that will keep you safe as you go up into the tree. And I didn't know, or I had done this a few times with a friend in Honolulu who was an arborist who taught me a little bit, but I hadn't never climbed really big trees before this project. And so the people at this biological station taught me how to do this along with my friend who was also climbing. And so once the line's up, you then inch up the line using a hand ascender, so that kind of grips on the rope and you use your hand to get up, and then a foot ascender, which you can see on my foot there. So you kind of are doing this hand and foot climbing up the rope, and you're wearing a harness as you go up. And it's pretty fun. It takes a while to get all the way up to the top of the 100 or 200-foot tree to collect data. And so I worked the entire summer with Jen Schlauk, who's on my left here, this woman, and she is an entomologist. And so she studies canopy ants. And so we worked together all summer. That she would be on the ground watching my safety and helping me while I studied the orchids. And then I would come down and she would go up into the tree and then she would do a survey of ant diversity.
Robert.
In the tree. And so, yeah, she was a great friend and we worked together all summer, and we really couldn't have collected any of this data without her. You really need a climbing buddy to climb trees with. And so, yeah, here's some of the pictures of us moving up into the canopy, or of me, that's me on the right. And I would have like my clipboard, and I'd usually be up there for between like 3 to 4 hours, two to 4, depending. And so I would have like a snack and my clipboard with my data sheet, and then all my measuring tools and some water, and you'd head on up to be up there for a while. It's a picture that Jen took of me up in the top of a ceiba. And so once you finally got to the top, you kind of find a branch that's good to sit on, and then you get yourself situated in a place where you can see all the branches and see all the orchids, and then start to use binoculars, or just if you're close enough to your eyes, just to identify them and start writing down information. So this would be the view once I was up in the tree. This is me sitting next to some maxillarias. Here you can see a whole beautiful bank of them on this high branch. And so this is like 90 feet up in the air, and this community of maxillarias is just doing great. And then on the right over there, I don't know what species that is from here, but there's also just the kind of the views. You'd look off down the branch and see all different types of orchids. And so you can see in this situation how having slightly lower diversity would actually help you, because without a flower, as many of you know, it's almost impossible to identify an orchid to its species. And so many of these didn't have flowers because they only have flowers, you know, for a few days a year potentially. And so having low diversity helped me make accurate identification. Since there were only 35 potential options, I could often use leaves or bulbs or other characteristics if I couldn't see a flower to identify it. Now here's more scenes from the canopy. So there's a Sobralia on the left. Sometimes species of Sobralia. Oftentimes I only got to genus, because if I couldn't find a flower, I couldn't get it effectively to species, so I just leave it at genus. And you can see they often grow out of these beautiful moss mats, which I'll talk about in a little bit here, about the importance of the moss in the canopy. And so you can see branches. I would look down sometimes if I couldn't. I would try to look, or I was trying to identify every orchid in the tree and count their numbers. And so I would try to get to parts where I could see branches from different angles, so I could make sure that I was seeing every angle and not potentially missing an orchid. And of course, there is some degree in error in this. They're huge canopies. It could stretch, a branch could go 50 feet in one way and 50 feet in the other direction. And I can only move a little bit walking along the branches while I'm harnessed in. And so there is some degree of error that I probably didn't see all the orchids up there. But you know, you do what you can, and you just describe that very clearly in the methods when I write my article, and it is contributing to the general understanding of what's up there in the canopy. But the canopy really is a frontier and a mystery. It's difficult to access, and not much is known about the ecology of these arboreal spaces. More beautiful scenes from the canopy. You can see these ferns just dripping off of the branches. So orchids going up, ferns going down, just like thousands of individuals. Really beautiful. More orchid pictures. So this is all just kind of tropical forest, beautiful orchids. And there were often monkeys up in the canopy as well. The monkeys were a little scary. I didn't really like them too much because they would be kind of aggressive. Sometimes they were very peaceful and they would just hang out in the tree or on different branches. Other times they'd be kind of aggressive and would kind of scream at me or get really close or play with my ropes. And that was not nice at all. So if the monkeys became too aggressive, I would just leave the tree and go down and come back another day, since it's their space, really. they're very agile and can be very alarming when they scream. So throughout the summer, I climbed a total of 34 trees of 9 different species. So the host, I'd climbed 9 different species of host tree, because I was also trying to see the effect of host tree on the orchids themselves. So the different types of trees have different communities of orchids. And so 34 generally in ecology, you'd hope for a sample size larger than that, but with climbing really large trees, it was hard to climb lots of them. And so I was, fairly satisfied with 34 by the end. I think when I first started the project, I had aspired to do 100 over the course of the summer, but as the reality of how hard it was to climb them set in, between getting the line in a safe place and hiking out to the trees, finding doable trees, I did 34 in the end. Usually I could only climb 1 to 2 if I was really going for it, 2 trees a day. And you have to choose a really good weather day. And it takes usually about 1 to 2. You can only do about 2. You can only rig 2 trees a day, and then you can only climb 2 trees a day. So it really kind of take a while. This is all within about a 4-mile area. And so you can see here's the research station down here where the red roofs are. This is where I was living with other people at the station. And then we'd walk out every day, Jen and I, and to the different trees and work on climbing. And so in the end, I found 35 species were found in the canopy, and here's some of the different pictures of them. We've got a Lockhartia down here, a Trigonidium, a Scaphyglottis, Maxillarias, Dressarellas, Epidendrum. So you guys might recognize some of these genera, at least some of these types of orchids, some of which have been integrated into the horticultural trade, and hybrids are now made out of them. I don't know so much about that whole end of things, but some of these are the base species orchids from what some of these hybrids arise. And so one of the questions was, so what would be the relative abundance of the various orchid species at Piro? And so, which is basically asking which species are rare and which species are common. And so this graph kind of tries to show that a little bit. So this is the results of my survey. So I made N equals, so I made 2,640 observations of orchids in Piro. And this is that block color thing on the left here, is showing of all those orchid observations, how does that break down into the different species? And so you can see that they're not evenly distributed. And so it's not like I would find one of each of the 35 all the time. Some are much common and some are rare, as you would expect in a forest system. And so you can see I found 852 Specklinia microphylas, while I only saw Cloecia wartzichii once. And so this kind of shows the breakdown of which ones were very common. So we've got Scaphyglottis, Maxillarias, Anathalis, Camaridiums, all very common, would see them all the time. And then quite a few that I only saw once or twice. And so those are not quite as common in the canopy. The Jachinella, the Aspasia, some of the Proseccia abbreviatas were not so common. So this is interesting and good information to know for management of epiphytic orchids. First, you have to know which ones are rare and which ones are common. And so here's some examples of the common ones, as a Scaphyglottis and a Specklinia. Those were just about everywhere in the canopy. And then, so then there's a Cloecia and a Jachinellia that I only saw once of each of those. And that was very exciting to make that identification. Okay, so some more results answering the question of what is the vertical distribution of epiphytic orchids in Piros. This is wondering where, as you move up a tree, are the highest density of, this is just abundance. We're not looking at diversity, just abundance of orchids. And I also put ferns on here. And so you can see that I didn't find a single orchid ever in zone 1. So the lowest section up to about 10, 15 feet, never found an orchid there. Moving as I moved up the tree on the trunk, would find a few. Then we would enter the area of highest abundance here in zone 3. So that's the branching zone, the first branches of these big, gigantic forest trees. That's where all the orchids usually were found. But then there's other species that were found then moving up into zone 4, and then the apical tips of branches, a few of those as well. You can maybe you should also note here the correlation of abundance of epiphytic ferns to epiphytic orchids. So the highest abundance of ferns also occurred where there was the highest abundance of orchids. So both of these epiphytic life forms are living in the same part of the canopy. All right, so this is another part of my results. This might be a little confusing, but this is an NMDS, so a non-multidimensional scaling of the data points. And so basically what this is showing is the different communities of orchids that were existing in the canopy. And so each of the dots is representing a community of species that I observed. And then the arrows are representing how those communities are separating. And so what's kind of driving, not driving, but what's separating those different groups. And so that might be a little confusing, but what's important to think about here is that the different communities of species in the canopy are separating based on Johansson zone, which is place in the canopy cover, so amount of light that they're getting, and the presence of bryophytes and pteridophytes, so ferns and mosses. So this would make sense to people who are excited about the horticulture and growing orchids, that the amount of light that they get, the place that they are, and the presence of other ferns and mosses is going to be very important. That those are going to be driving variables for the success and diversity of a wild orchid population or an orchid that's growing in your house, in your yard. And so one of the results that we could kind of take from this is that there are certain orchids that live in open or exposed areas. So these are the communities of orchids that are preferring habitats at the tips of branches, very sunny exposed locations, or on types of trees that have open canopies that are going to allow more light to get through. And so some of the examples of those species were Nidema, Caularthron, Brassivola, and Polystachya. And so then other species in the canopy at Puro are going to be shade tolerant or shade orchids that I only ever found, in these mossy little areas close to the trunk, or in shaded areas underneath dense foliage, or in areas with lots of other pteridophytes and bryophytes or other ferns and mosses. And so those examples were Maxillaria, Lockhartia, Sobralia, and Specklinia. And so this result kind of shows that it's not just uniform what's going on up in the high canopy. There's micro niches, so little pockets of places where certain species of orchids have adapted to thrive and to grow. And so the canopy is not homogeneous. It provides a variety of small niches that these different species have been adapted to live within and to live with each other. So here's a different visualization of that same concept. So going out along a branch, so the base of the branch closest to the trunk, you would have your shade tolerant, wet little community of Sobralias and Lockhartias and nice little flappy leafed, thin leafed things. And then as you're going out on the branch, it gets more and more exposed, hotter, less moss, less ferns. It's a newer part of the growing tip. And so it's fresher bark. So it's not as, it's potentially less textured to hold less moisture as the older parts of the same branch. So then as you move out, you're going to get your Nidemas and Caularthrons, Dimerandra, and Polystachya. And so there's differentiation along a single branch. And this is really fascinating that this branch might be only 30 or 40 feet long, but you're going to have different communities of orchids living within it and on it. So I drew this to kind of show this this concept. It's not a scientifically supported drawing yet, but I'm still working on the analysis part of this project to kind of back up some of these things I'm saying. Moving on. And so a final bit of the analysis I'll try to share with you guys today is I use these models called structural equation models to figure out which variables are driving orchid abundance and diversity. And so this might look really messy and confusing, but what it's basically showing is that there are a variety of established variables which influence orchid fitness. And you folks will probably know this, right? That precipitation, so water, will influence fitness, humidity will influence fitness, temperature, elevation, all impact whether an orchid will do well. Moving beyond that, we have social, in a forest setting, we have social drivers. And so what happened to the land Historically, 100 years ago, 200 years ago, 2 weeks ago, will have impacts on the habitat for the orchids. Of course, if they're getting harvested, that also impacts them. Over on the right, we have host characteristics. And so that's the tree that the orchid lives upon. So the different characteristics of that tree. So nutrients in the bark, the size of the tree, the age of the tree, the density of the leaves in that tree, bark texture, phenology is when it flowers or when it loses its leaves. And so those are all also going to impact a orchid living within that tree. And then we've got some other biotic ones, so bryophytes and lichens, whether or not there's surround, and then mycorrhizal fungi, of course, which are, I didn't look at mycorrhizal fungi at all in this project, because that would be a whole nother project, but they're very important for, as we all know, for orchid establishment and growth. And so what this diagram is trying to show is that these variables all influence orchid abundance and diversity, but not just directly, they also they work in a network. And so perhaps precipitation is going to impact the humidity, which then impacts orchid diversity, or humidity is going to drive the distribution of mosses, and that's then going to influence orchids. Or maybe the canopy cover of the host. So if it's a fig or a ficus, maybe, or banyan tree, the density of the leaves will then impact the humidity, which would then impact orchid diversity. And so it might not be a very intuitively diagram. It's just showing that these variables are interacting with each other to influence orchid abundance. And so there's a lot of studies that have looked at the individual impacts and correlations of each of these variables on an orchid's abundance and diversity in the wild, but there's not too many that try to do interactive effects with these structural network models. And so that's what I am in the process of trying to do, is to use my data set to try to make a network model and to test these different networks to see what is interactively working on driving these communities. And so I can show you a preliminary result of this. So I kind of distilled all of those factors into just these 5 variables that seem to be most important in Costa Rica. And so those variables were location in the canopy, surrounding bryophytes or mosses, pteridophytes or fern density, surface area, and canopy cover. All of those were the most important within my data set. And among those, the most important positive correlating factors were bryophytes, pteridophytes, and orchids all had this kind of like positive feedback loop. And I'm still working on the analysis, so I can't like totally say that this is a significant result yet, but it's looking like the presence of a moss layer might then facilitate the presence of thick ferns around it on a branch, which would then positively facilitate orchid abundance and growth. And so this is really interesting because it's showing this kind of cascading effect of one organism allowing mediating an environment or kind of changing it so the next one can then be successful, which will then allow for the orchid to be successful. And so these are the models that I'm working on now, and hopefully we'll finish so I can for my dissertation. And yeah, and so in conclusion, my preliminary conclusions for this research was that epiphytic orchids in Piro in Costa Rica are not homogeneously distributed, but are heterogeneously distributed. So they're unevenly distributed in the canopy, and that varies based vertically and between different host species. And there's evidence to support the hypothesis of habitat facilitation cascades between moss, ferns, and orchids. And finally, that there are orchid communities of open and closed canopy groups in Piro, which are differentially influenced by moss and fern layers. And so this were kind of the results of my working in Costa Rica for the summer. And I definitely hope that I can go back sometime and ask more questions and get to climb more of those trees. It was a really beautiful experience to get to learn how to climb, to get to spend time up in the canopy, which is just one of the most beautiful environments I've ever been in, and get to become familiar with this set of orchids and ask questions about why they are in certain places. Yeah. Oh, and yeah, I'd like to say thank you to all the people in Costa Rica who worked at the field station and who helped me work. Jen, the people who helped me in the field, and the other people who were my friends throughout that period at the Piro Biological Station. And so that's all I have for today. The people who've been funding my project, thank you to all these different organizations who've been funding my different projects through the last few years. And yeah, huge mahalo to the Honolulu Orchid Society for having me speak again. I really enjoy speaking to you guys. Yeah, and hope to be in person next year. And we'll be really happy to try to answer any questions you might have either about Oaxaca or about Costa Rica.
Hi, Julia. This is Kate Leonard. I was just wondering, did you learn about the species in Costa Rica after you got there, or was that something that that you knew and chose that project because there were orchids you were already familiar with?
No, I wasn't familiar with those orchids before I went there. I mostly just chose it because this organization was offering a 6 or 7 month fellowship to live at the field station. So I applied with my project, and yeah, just have to kind of follow the funding that's available. But once I got there, I learned with help from people there, the orchid species, and worked on getting good at identifying them.
Well, it's very amazing, and I'm sure everyone would agree that it's just spectacular of you to share this with us. Thank you.
Thanks. Yeah, thank you.
Julie, I want to thank you for your presentation. I am blown away. It was so good. We had talked a little bit earlier about the circumstance which led you to Costa Rica, and you were saying that your work in Mexico was being restricted because of COVID and you couldn't travel there. And then this Costa Rican fellowship became available, and it actually allowed you to travel and stay in one place that didn't have a lot of people. So that all kind of worked out. But the work is fascinating. There's so many things going on. And obviously, it's not easy to do what you do. So that's why we don't have as much information.
Yeah, thanks, Brad. Yeah, well, yeah, on this part about needing to go to Costa Rica instead, especially because my project in Mexico is really based on this big celebration and this big kind of in-person event in which they have big parades and they do these big celebrations. All of that was canceled for 2 years. And so no one went out and collected orchids or did any decorations for 2 years. So that's why I couldn't really go there. There wasn't really anything to do. And because it's such a social project there, I have to interact with a lot of people. So it wouldn't really have been right to try to go and do a lot of in-person interactions when COVID was a problem. And in Costa Rica, it wasn't really a social project. It was just me and Jen out in the forest with orchids. So that enabled it to be a little bit more feasible for the pandemic. Yeah.
So I have one question regarding the Mexico work. If those orchids grow well on trees, and I think you told us before that there are certain species that the orchids prefer, can those same orchids grow more in town, or do they really need that environment out with the trees?
People grow them in town. Yeah. So some of the people, especially the harvesters, will take some of those pseudobulbs and bring them back to their gardens and courtyards and patios and grow them there. But they take a lot of care in those environments. So you couldn't really just leave them on the tree there. You'd have to water them quite a bit, and kind of, it would be more of a horticultural situation. Whereas in the habitat that is suitable to them, like in the place where we transplanted them, you could just leave them there and they'll do quite well, hopefully, without too much care. Yeah. I mean, I've seen people put them on like plastic Coke bottles, or growing them on metal. I mean, they're pretty vigorous if you water them and are, as you folks know too, like you can grow orchids on a variety of like materials. And if you're attentive, they can do well, but they will thrive if they're on their correct host species of tree.
Julie, I was wondering, you said you saw monkeys up in the trees. Did you run across any other animals like snakes or anything in the canopy?
Not in the canopy. I was always afraid of seeing a snake and never did. So that was good. But we did see lots of very poisonous snakes on the ground around Costa Rica, because there's terciopellos, which are fer-de-lance snakes. They are all over the forest floor there. And so it was more concerned like getting to the tree. And then, but once we were up in the tree, you were kind of like safe from the fer-de-lances. And so,
Okay.
Yeah.
Interesting.
So Julia, when you talked to us a couple of years ago about your Oaxaca, I'm sure I'm saying that wrong, project, at that time, there was a horticultural aspect of it where you were collecting it and you were trying to grow them, not in a greenhouse per se, but it was almost like you were mounting them to fencing, or you were trying to cultivate them and then reintroduce them, as opposed to just picking the most viable pseudobulbs. I was wondering how that worked out.
Yeah, it worked out well. The main purpose of trying to grow them on that fence, like you're remembering, is because we were just trying to see if it was even possible for this species to grow from single pseudobulbs and like produce roots. And kind of, if once we had the information that it was possible, we then moved on to just sticking them on trees. But I do think that it would be a potential solution in the future to grow them for a while. Or this is kind of up for debate, is whether or not to just immediately put the bulbs onto the tree, or to try to cultivate them in a greenhouse for maybe a few months first. And then once they're more established, then moving them. It's unknown what would cause more stress, moving them once they have roots, or putting them into the forest when without any roots. And so that could be something we experiment with in the future. Something kind of detracting from the idea of working, and then is because there are no nurseries in this town that would really be suitable for this. And so trying to make it as simple and as economically feasible for people there would just be to immediately put them onto the trees and not really worry about cultivation too much.
Thank you.
Yeah.
Hi, Julia. I was actually wondering because I wasn't here 2 years ago, so I was just wondering what really got you interested, and what positions did you really try and find at UH that was able to help you get into this position?
Yeah, I started as a master's student in Tamara Tickden's lab, I'm still in her lab at UH in the botany department. And I just really wanted to work in anywhere in Central America with any plant. And Tamara was working with orchids already in Oaxaca. And so she kind of sent me to Oaxaca, or kind of honed me in on Oaxaca as a place that has a history of orchid use. And she had already been working and had collaborators there. And so that's how I initially got interested in it. And once you get really excited about orchids, especially, or any kind of a specialty, then you just get more and more interested and excited about it. And so I started as a master's student, and I got a few small grants that kind of helped me go there one summer, and then the next summer. I'm not really in any, I don't really have a fixed position or anything. I'm just still a grad student, and I'm kind of funding my dissertation through a series of small grants. And so one day I hope to get a real job, but I don't have one right now.
Yeah, it's very interesting. I just want to one day maybe go to school for something like that too. It sounds very an interesting career choice, and just it's just fun, it seems to me.
Yeah, absolutely. I think a big component is finding an advisor who's really supportive of doing international projects and wants to support you, not financially, but conceptually support you to kind of follow what you're interested in. And then after that, just looking for grants that are offered by various organizations to find your own money, because find money for the project. Because once you have that, then you're kind of an independent grad student. You don't really have to be too dependent on staying at UH or kind of doing someone else's project. You kind of can do what you'd like. So yeah, it's very possible research things you're interested in, especially orchids. Yeah.
Thank you. Thank you. Actually, I have one more quick question about something else too. I was wondering because when you said that orchids, a lot of them can grow in like communities and clusters. I was wondering because I know some other types of plants, like when they are clustered up, they usually tend to fuse a little bit, right? So I was wondering, can that also happen with orchids too? Like, are they somehow able to fuse into a bigger plant?
Like multiple individuals kind of growing together.
Yeah,
Uh,
like
I think when I was observing things, I wasn't observing them through time. I would just see them once when I climbed the tree. And so I couldn't tell if I just saw a thicket of orchids. They look like thickets kind of. I couldn't tell if that had been multiple and they had kind of grown all together, or if it was just one and then had slowly grown. So, I mean, there's some interesting questions that would come out of observing a population for a long period of time. So I don't know if two individuals can fuse their roots. I guess I haven't observed that, but them clonally reproducing, so a single orchid getting thicker and thicker and thicker on a branch, and then parts dying and then parts growing again, I think is a possibility, but I don't know too much about that.
oh, thank you though for the information. Yeah.
Yeah, right on. Yeah.
I have a question, Julia. This is Linda, Inoue.
Hi.
What's your childhood like? Did you have a childhood interest in botany or in plants of any kind?
Yeah, I think I always was really into plants. I grew up on like halfway up Round Top Tantalus, and so I spent a lot of time like on all the trails up there and hiking around and poking around in our yard, and really liked, I guess, plants. I mean, growing up, I didn't really know that a lot of the plants in Hawaii are invasive, and so like our whole yard was totally invasive, and I just thought it was great for a long time. And it was kind of like later, once I started studying botany, that I realized like, oh, none of those are good plants. But yeah, I guess I first got really into botany through getting excited about native Hawaiian plants. Like after undergrad, I did a few like temporary technician jobs working with plant conservation in Hawaii on the Big Island and Kauai. And then from that kind of wanted to do a botany degree. And then from there wanted to study orchids. But I think in the long run, yeah, I'd like to study Hawaiian plants probably and come back home predominantly.
Thank you.
Mm-hmm. There's also, this is a side comment, but we do have like a really interesting flora of our own native Hawaiian epiphytes. I mean, we don't have too many epiphytic orchids like you guys know, but we have really amazing and diverse epiphytic ferns that live in our cloud forests. And so that's something that I'd really like to study next, is epiphytic ferns up in the ko'olaus or on other cloud forests throughout the state. Not too many people study them. There's only a handful of people who are really interested in looking at the epiphytic ferns of Hawaii. And so that's something I am interested in.
Julia, are there a great number of those ferns?
Yeah, we've got like maybe as many as 30 or 40 species that are endemic, so native to Hawaii, of native ferns. They grow up in the ohias, like sometimes there's so many of the ferns. Maybe some of you have probably seen it on hikes up to the summit ko'olau areas, but like so thick that they'll overcome the ohia tree almost, just like loaded and those are in the genus Adenophorus and Elaphoglossum. Yeah, so we have some beautiful epiphytes.
Julia, I was really intrigued with the climbing that you did, because I don't think many of us can do that. What kind of training did you have to do that kind of climbing?
Probably not too much, really. I had climbed with my friend who's an arborist around Honolulu. We had like climbed around the city a little bit, and then after that, I just kind of got there and started doing it. The first few times that I climbed, I was really quite scared. Like when I got really high, Yeah, like, I mean, I remember the first time I climbed like a 150-foot tree, like my like hand was shaking, so I couldn't write anything down. And like, I was so scared, I couldn't really move it around or like do anything. And like, it was actually kind of disheartening. Like the first time I climbed under the trees, it's like, oh, this is really hard and it's really scary. And I don't know if I can do it, but I got used to it. And like, you kind of accustomed to being up high and just kind of not as afraid of just the space beneath you, and you kind of get more comfortable. And yeah, so you get accustomed to it. And then physically, like it's hard, but only very specific muscles, I think. And so like once you kind of get that weird muscle set, then you're good.
Did you have any near accidents?
Yeah, we did actually. I think that I would like more training because there were some scary instances, like once having the rope, it was kind of an optical illusion that it looked like the rope was going over a really big, thick, like huge branch, but it was only actually going around a branch like as thin as my wrist, maybe. And because that is kind of hidden, just like the way the rope looks, it's hard to tell sometimes. And so like I got all the way up to the top and then saw that it was on a really thin branch, which is very scary. And then went all the way down again. Then you like take everything apart and then reshoot the line and everything. But yeah, there's some scary things. I mean, it's hard to have, it's like an inherently risky activity. And so, yeah, but I think I hope to get trained and learn more so I can be safer. Yeah.
And Julia, it looked like the area was pretty isolated. So if there was an accident, it would be pretty difficult to get help.
Yeah, I think that's true as well. It was like an hour and a half down a dirt road to this station, and the nearest town didn't really have a very good hospital. So yeah, there were a few factors that would have gone bad, but luckily nothing went bad. And I mean, we tried as hard as we could to make things safe, and we weren't really trying to take risks necessarily. We were just trying to get our data as safely as possible. It wasn't like a thrill-seeking activity, although it was thrilling. That wasn't the intent. That wasn't the point. Yeah.
So was there synergy between the ant study and the orchid study, or you were just in the same place at the same time?
We didn't plan it together. Like they picked the two of us to get the fellowship separately, and then we kind of became friends and ended up working together. Really luckily. But interestingly enough, Jen and I would like to write some kind of paper together, or like look at correlations, because there were interesting correlations between ant diversity and orchid diversity in the canopy. And that kind of data set of having like someone looking at botany and insect diversity in a canopy is pretty rare. Like usually just like one type of interest gets focused on. And so, yeah, we plan to both do that in the future. We're both grad students, and so kind of moving slow on that. But yeah, it turned out actually that the areas that were higher in orchid diversity, so these wet, shady areas, actually had lower ant diversity. And the ants really liked these like sunny open areas where there would be lots of species and they'd be running around looking for food or something. And so, yeah, there's some interesting things that we found for sure.
Do the concheros have any kind of rules or regulations that they need to follow when harvesting the Prosechia karwinskii? Or are they harvesting every single yellow flower that they find? Or is it something to do with like maybe it's a minimum number of pseudobulbs on a plant before they can harvest?
Yeah, that's a really interesting question. It depends who you ask and like some of the older or some of the orchid harvesters will just take every single flower they see on a tree, and other ones are kind of more aware of the sustainability of populations and will only take half of the pseudobulbs. And so it kind of varies between the different ones. I think there's a little bit of like an educational gap between understanding that the population of orchids comes from the flower structure. Like I think a lot of the people who are harvesting orchids kind of think that they're clonally reproduced. They just kind of grow from each other on trees and don't realize that the impact of taking an entire flower, especially for a orchid that only blooms once a year, because if you return to a population every year and take every single reproductive flower of that population, you're just the number of seedlings you'll get will just go down and down and down and down, which is what's happened. And so I think kind of that educational piece would hopefully do a lot. So no, they don't really follow too many regulations right now. Like as I was saying, the decline of orchid numbers has happened really fast. And so it's happened fast enough that they haven't really responded by creating a sustainable strategy. Like what used to be fine in the past has changed within a generation. So like the grandfathers of the current concheros, they used to be able to take thousands and thousands and thousands with there being not really any impact, or they were able to do that for quite a while. At least there probably was an impact. So yeah, it's I think the tradition and the population of this plant are both in times of active transition. Like things are actively changing within the last decade or so. And so, and then from a federal perspective, harvesting this orchid is entirely illegal from a Mexican government perspective. Like it's federally protected. It's on the endangered species list, but that's just kind of broadly disregarded by across, and that's disregarded by many communities with many species of orchid, which just kind of reflects that like an effective strategy can't just be like a federal blanket ban. It has to be like a community-based conservation plan. Yeah.
Are there orchid nurseries in Costa Rica or Mexico?
Yeah, there are, but yeah, I mean, people are growing like in vitro, growing large quantities of species orchids in Mexico. I'm not super familiar with the people involved in that, but the main kind of problem, at least for Oaxaca and conservation, is that the people who are in control of these plants are these really rural, small communities with not a lot of economic resources, and they can't really have a in vitro success of like big orchid greenhouse. And so unless there was like some kind of neat project in which, you know, propagules were sent to the labs in bigger cities and then they were sent back to plant in the forest, and that would be really great. That's kind of beyond my ability to facilitate, but I think that'd be great if that was set up. Yeah. And in Costa Rica, I'm not sure what the people love orchids there, and there's a lot of growers and a lot of greenhouses. I guess I've never heard of a project which climbs into the high canopy to plant orchids, though. That would be really neat. I'd like to do that, but I don't think anyone's doing that right now. People are kind of focusing on the lower forests, focusing on horticulture, kind of not in the canopy.
So Julia, in Costa Rica, in the canopy, it looks like all of those orchids are sort of endemic or happen naturally. How do you think they got there?
I mean, I think they kind of evolved to be part of that system. Most of the species that are there are common and widespread. And so almost all of the 35 species I was studying have distributions, a lot of them that go all the way from like the southern US all the way down to Brazil. And so they're big, wide distribution, common species. And the seeds of these species are probably like so common that we're like, you're breathing them in if you visit Central America. Or if not that common, that at least that they get distributed quite a bit. And they're quite, they're like pretty scrappy species, like they're capable of living in a variety of habitats when the seed falls in a correct place. But beyond that, I don't know if I'm not qualified to speak too much about the evolution of orchids. I'm only able to talk about ecology and distributions slightly.
It was interesting where you didn't observe that many that were lower to the ground. Most of them were kind of caught in that first area of branches.
Yeah, that's something I've wondered about too, because it's like there's nice little mossy nooks at eye level or about 1 foot off the ground, and I never found anything down there. I don't really know why. It could just be quite shady. It's just like too dark. It's quite dark down in that section of the forest. Yeah, that's a good question.
Okay, we'll take one more question. Thank you very much for answering our questions. Your work is fascinating, though. Any more questions? Go ahead and unmute yourself.
For the project in Mexico, does your restoration strategy have any way to deal with maintaining genetic diversity?
That's a really good question. And that's one of the biggest criticisms usually about this restoration plan and what I'm doing is that there's not enough attention paid to genetic diversity because the concheros, the orchid harvesters, go out across the entire state to harvest this species. And so it probably is only this one species, like they're not mixing species, but they are getting it from all these different populations, which have genetics, which are, adapted to those different areas, slightly colder, slightly wetter, all these different genetic variations. And no one's done population genetics on this species. So we don't really know about the existing diversity. So these orchid harvesters are bringing all these back. They're mixing them when they create these big decorations. They're getting thrown around and put in boxes and moved around. So by the time they have arrived to me, I don't have any idea where they've come from. And then I'm just reintroducing them in a big patch of mixed genetics. And so this is a somewhat problematic practice. And I acknowledge that, but the only real response to that is that if no one was growing them, they would just be discarded and they would just be thrown away. And there aren't too many instances of mixing population genetics really resulting in negative impacts on the genetics of a species of orchid. And then the final idea is also that this is not really a truly wild place. Like I'm not introducing them way back into a pristine preserve of orchids. This is more of an inter situ site. And so there's like a farmhouse, that's quite close. This is kind of a place where people come and go a lot. It is a forest of adequate trees, but it's not a preserve by any means. It's like a mixed use landscape. And then the idea also is that this population of this like weird Frankenstein mix of population genetics will also be, if it's successful, a place where the people of this town can go in the future and harvest from this site. And so it could be an inter situ kind of harvest site with mixed genetics, but that hopefully won't be too much of a problem. And then the other good response too would be maybe I should work with someone who could do the population genetics and we could learn more about this species.
Thank you for staying and answering our questions. If there are other questions, please do send them to me and I might be able to pass them along to Julia. We want to thank you for your outstanding presentation. It makes us think a lot and makes us much more aware of conservation efforts, which will have a positive impact in the future, hopefully. And we're also very excited to hear about any future work that you might have, either in Hawaii or other places, whether orchids or ferns or anything else. Everything is very, very complex and interesting. So thank you.
Yeah, thank you, folks. It's so great to talk to you. I really enjoy talking to the Honolulu Orchid Society. It's a very enthusiastic group, and it's really great to talk to people who are super, honestly enthusiastic about plants and growing them. So thank you, folks, and hope to see you when I'm home in a year in person. Yeah.
Yeah. So thank you.